So last class we talked about the cell cycle. This year's the cell cycle, and we talked about how the cell cycle has mitosis, G1, S, and G2. G1, S, and G2 are part of interphase. Just remember that's where the cell material is being replicated. So interphase, this yellow portion here, is cell replication. If you look at M phase, that's mitosis. In order for the cell to actually divide into two identical cells, we call them daughter cells, the organelles and the DNA have to replicate, and then it can divide into two identical daughter cells, pictured over here. Oops. So we talked about how G1 was, and G2 was where there's proteins that are made. And we also talked about how G1 and G2 had organelles. Replicated. So remember that organelles are subunits within the living cell that actually have function. Well, today we're going to focus specifically on S phase. So remember in S phase, S stands for synthesis. Specifically DNA synthesis. And that's where the DNA is actually copied. So remember, our whole objective in the cell cycle is to make two identical daughter cells. In order to do that, we have to first copy the organelles, which we did in G1. And then in S phase, that's where the DNA is actually replicated. Once we have the DNA replicated, we can go on to G2. And then in mitosis, that's where cell division occurs. Remember, not cell replication in mitosis. That's where cell division occurs. So in S phase, the cell synthesizes its genetic material so that each daughter cell has identical copies. Let's write that down. The cell synthesizes its genetic material. Oops, no apostrophe. Each daughter cell has identical copies. So what do I mean by that? Well, let's say you have a skin cell. Well, there's two things I want to touch on. I want to talk about what a daughter cell is and what's actually happening in S phase. So let's say you have, oops. Let's say this is a skin cell. Let's say this is a skin cell. And let's say this skin cell has a nucleus. That's in red. That's where the genetic material is stored. Alright. Now let's say you have this, there are other skin cells that were damaged, and now you have this healthy skin cell over here that wants to replace those skin cells that had been damaged. So this skin cell here is replacing other skin cells in our body that have been damaged. So this skin cell has its genetic material inside the nucleus. It has 23 pairs of chromosomes. Let's see if I can draw this. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23. Whew. Okay. Alright, so let's say that's 1, 2, 3, 4, 5, 6. There we go. So before this skin cell can make an identical copy of itself, it needs to first copy its genetic material. So far, before it's copied its genetic material, it has 23 chromosomes. And we know that because each chromosome has a knob that we call the centromere. And let me draw this, kind of hard to see. Okay. Oops. Almost there. So if we count each circle or each centromere, these are supposed to be circles. We have 23 chromosomes. We also start off with 23. I'm sorry. Oops. I am sorry. That's not 23 pairs of chromosomes, I meant. Or we start off with 46 chromosomes. So if we count each chromosome, we count the circle, which is a centromere, And there's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, all the way down. And there's 46 chromosomes. You can also count the leaf-like structures, which is, this is counted as a chromatid. To make intuitive sense, we're going to count the cells by chromatids, even though it's not biologically correct. So each chromatid is each leaf-like projection. So what do I mean by that? This leaf-like projection, this single leaf-like projection, is a chromatid. So let's actually take one of these and pull it out right over here so we can see it a lot better. So she draw a centromere. So this is before DNA replication. So before DNA replication, we have 23 pairs of chromosomes. Actually, let's not even talk about 23 pairs at the moment. We have 46 chromosomes. And 46 chromatids. So after DNA replication, actually, I'm out of time. We'll continue in the next video.